briefly, as I understand it, the goal here is to sort of explore how animal data might be useful in terms of understanding sleep. So I want to provide a very high level overview of sort of how we think about what's going on during sleep in the context of memory, something that we think it's quite important for. And then I'll talk a little bit relating to things Michael said and also things Jonathan touched on about, what sort of things the animal data might provide. And to start with that, I think it's important to have an initial sort of model, or how are we starting to think about these processes? And if you think about something fundamental like memories, right? And I'd like to point out, whenever people are at a talk like this one, the point of you being here is to make memories. And if those of us who are speaking to you fail to drive plasticity in your brains and fail to create memories in your heads, then we have wasted your time. And we also know, though, of course, that after an experience like this, that there's a lot of post-experience processing that goes on. But during an experience, right, we have the sensory inputs that you have going on right now as you see and hear me, and maybe you're smelling your coffee or whatever else it is. We know that those in the brain are coupled to a wide variety of sensory and motor circuits, which are in turn coupled to sort of a host of maybe more higher order circuits like the prefrontal cortex and so on. And being a hippocampal person, I'm sort of required to put the hippocampus on the far right-hand side of this diagram, but there's not for no reason at all. As many of you probably know, the hippocampus is utterly critical for forming memories for the events of daily life, like this webinar. So without this, you could listen to what I said, but you wouldn't have any memory of it 5 minutes from now. And so this is incredibly complicated because everything in the brain is connected to everything else. And, just as Jonathan was talking about circadian rhythms and how those might be entrained across all of these areas, we're studying neural activity patterns and how those might be flowing through these incredibly complicated networks and how that works. All right, so while you're awake, like now, hopefully, you are learning things and plasticity is going on. And the current thought is that is driving strengthening on average, or by predominantly strengthening of synapses throughout these structures. We really don't understand exactly where that's going on or how that's happening yet, which is kind of remarkable, but we know something is going on. But at the same time then, after you've learned, that means you've on average perhaps strengthened a bunch of synapses. And there's a fair amount of evidence that one thing that happens during sleep is some fairly broad synaptic downscaling that is basically erasing stuff with the idea that this is setting up the system to be able to learn again and maybe to filter out unimportant stuff. Where this happens, how strongly it happens, I would say is largely unclear. There's some evidence for it, but it's really not clear. And at the same time, then, we're just going to move that off to the side, we also seem to have active processes that help us remember important stuff. And so this idea has been that the hippocampus, which is a site of rapid plasticity during experience, allows memories to be formed. And then afterwards, during offline periods, both during wake and particularly during sleep, it seems to replay memories. And basically that means information flows, and I'll talk about how it flows, but it flows around this circuit in such a way that we think the hippocampus might act a bit like a teacher that is repeating patterns over and over again and helping ingrain those in the rest of the network. And so we have already an extraordinary level of complexity, synaptic downscaling on one hand, something that might strengthen individual patterns on another. This has to be presumably allocated to the specific areas that are important. So if you see and hear things, maybe visual and auditory areas need to be engaged. But if you feel or smell things, maybe it's somatosensory olfactory areas. And we really don't have a clue of how all of that works. All right, so the advantage of animal studies is, particularly with the technologies that we and many others are developing, we're starting to be able to sample activity throughout larger and larger parts of the circuit to actually understand how information flows. And I'm going to just talk about one result from a few years ago, which kind of gives you a sense of how that might work. But to give you an introduction then to that, I want to talk about two questions is, first of all, what does it look like when we think the hippocampus might be teaching the rest of the brain? So I'll show you some work on hippocampal replay to give you an idea of the kind of data that it's possible to collect. And then secondly, what is the flow of information during this process? And again, I'll try and go relatively quickly. So let's start with this first one. So what I'm going to show you here, and hopefully you'll be able to hear this because I think I shared sound. So this is the activity of 47 simultaneously recorded we think single neurons from the hippocampus of our little rat. So this is a top-down view of our rat in an M-shaped maze, which is shown here with this transparent overlay. And if you record from hippocampal cells in actually many organisms, you find that they are what are called place cells. And what that means is we can arrange the activity so that we get this sort of beautiful diagonal of activity. So each of these ticks is one spike, and then each neuron that is active in a particular region of space. All right. And so the idea here is that these spatial receptive fields form very, very quickly during experience. And there are actually a tremendous number or tremendous structure to these. You might see that there's sort of little bursts and gaps. That's something called the theta rhythm, which organizes activity and creates sequences of activity on very fast timescales. But the thought is that this sequence of activity or these patterns of activity are a sort of representation in the brain of an experience. All right. Now, this is what it looks like when the environment is simple. Whoops, sorry, wrong button. I'm going to show you an example with more cells. This is more recent data from an environment that's a little bit more complicated. But the reason I'm doing this is because one of the things that we can do with the animal data when we have enough neurons is we can decode it. So that means we can say what representation is being expressed at each moment in time. So in this version of that M-shaped made with this extra handle, so this is kind of like a trident, the animal's location where this event happens is shown here with this little sort of magenta dash, and this is where the animal's nose is pointing. And this green dot is the location where we think the animal is basically thinking about. And this color around it is a probability density around that of where we think the neural activity reflects. So in this case, you'll see the activity as the animal runs, and you can see that we can track that activity. You'll also notice that this green dot isn't always right on top of the animal. There's actually all sorts of interesting dynamics that happen while the animal runs around, where it goes ahead and behind and so on and so forth. But the upshot is then we can apply these same models to offline data, and that's both when the animal's awake, as I'll show here, and when the animal's sleeping, as I'll show in a moment. So here's an example of what's called awake replay, which is one of those events where we think the hippocampus is teaching the rest of the brain. So here's the animal, here's the position that's being represented, and you'll notice this will go by very slowly in time here, but there's a big burst of activity, and the mental position travels sequentially away from the animal over about 200 milliseconds and then jumps back. All right, so this is like a fast forward memory. We think this of as a memory consolidation event where the brain is recapitulating this pattern, and we can read that out based on the spiking. So it's kind of fun. We can do the same thing when the animal is in a rest box afterwards. So here's the animal just hanging out. That's the way its nose is pointing. And you see right at the beginning of this, the animal's we think the animal's representing where it is in the box. We start up the video, and then it jumps over to the track where it was some time ago, plays out this beautiful trajectory of going down, in this case, towards that well, and then stops. Similarly, though, we can also see even more complicated things. So here, we're starting in this other environment. The activity goes down, and then it jumps back to the box for a minute, and then it jumps to this back into the environment. So the point of all of this is to say that now that we have these higher density recording techniques, we can really get a sense for what the brain is doing at each moment in time. And we can ask, so is this the system integrating across memories, things that happened in this track and in the sleep box, for example, because these things are being replayed together? And the short answer is we don't know, but the data now exists to start answering these questions. Okay, so a few facts about replay before I go on to the second part of this. Replay favors important experiences. So we showed and others showed that rewarded experiences are replayed more, novel experiences are replayed more. So the idea is that the animal's brain, and presumably the human brain, has an active way to select stuff that matters and then have the hippocampus spit that out basically to the rest of the brain as, again, a kind of teaching signal. I'll just mention, those of us who show replay tend to show these beautiful sequential events. That's actually not the majority of events. Many of the events are just replay of a particular place or one place that hops to another place. So I just wanted to, give you a sense of what's actually going on in the data. These events, replay, this happens during something called a sharp wave ripple. The name doesn't really matter. This is just a signature that we can look for in the hippocampus of this kind of big burst of spiking. And it's been shown that these events can engage activity basically everywhere in the brain. So this first study by Logothetis et al. Demonstrated that in the primate brain, they could do fMRI and trigger it on the time of these sharp wave ripple like events in the hippocampus. And they found blood flow changes basically everywhere, with the exception of V1 for some reason, but almost everywhere else, thalamic areas, cortical areas, you see changes suggesting that this is really a brain-wide event. Similarly, we, as part of our demonstration of the flexible electrodes, Michael mentioned, we recorded from multiple cortical areas and striatum. All of these areas seem to be entrained, or a subset of the neurons are entrained by these events. And we're still trying to work out, what is that doing? How might that be ingraining memories? How is that engaging local circuits? And then the last thing, which I just think is kind of fun, particularly in a sort of sleep context, is we showed a while ago, and this was replicated later, that awake replay, that is the things that happen during downtime when the animal's just hanging out, is actually closer to reality than sleep replay. So sleep replay tends to include patterns that weren't necessarily part of the original experience. And this is kind of interesting from the idea of sleep as maybe an area where insightful learning might happen. So maybe it is the case that the brain kind of loosens the reins on what gets replayed so that it can make connections between things during sleep that might not be harder to do during waking. Again, fun hypothesis, not at all clear that that's what's going on, but that's why it's important for us to be looking at these data. Okay, then for the second brief half of this, I just want to talk then about the second bit, but first to remind you, so we have this idea of replay, which might be happening, which we think happens both during waking and during sleep, emphasizing these important experiences. And then I just mentioned sleep replay may actually include associated experiences. But again, we don't understand this, right? We have an idea that this is what's going on, but we don't really understand how it works in this sort of broad cortical engagement way. All right, so just to update this model then, we have this replay where the hippocampus seems to be driving activity through the rest of the circuit, both during wake, where it looks really nice and similar to reality, and during sleep, where it looks messier. And this comes back a little bit to the question that was asked about K-complexes and slow waves and so on. But it turns out that we've known for a while that these events where replay occurs, the sharp wave ripples, often occur after cortical down to upstate transitions. And just to make sure we're all on the same page, this is where you have a period of relative silence in the neocortex, and then it goes into a burst. So when you have a slow wave, the upward part of that wave, you tend to get hyperpolarization of neurons, very few cells active. The downward portion often corresponds to inward flow of current and a big burst of activity. And the sharp waves tend to happen after that happens in the cortex, leading the question of, who's teaching whom or who's talking to whom in these contexts. And that led us a number of years ago to kind of look at this and to try and ask this question, or get sort of the first little bit of insight into how information actually flows across these areas. And specifically, the question was, well, does replay really start in the hippocampus? And the reason this is important is if we think of this as what's important for forming memories or deciding which things get remembered, then it really matters who's calling the shots, right? If the hippocampus determines what's remembered, that's one thing. But if the neocortex determines what's remembered, that's very different. And we might want to think about how memories are formed differently in that case. Okay. Right, so there we are. So this is a study, again, done a number of years ago, but I'm guessing most people here are familiar with it, so I figured I'd talk about it. So this is from a former postdoc, Gideon Rothschild. And basically what Gideon was interested in is how sound information, auditory information, relates to hippocampus. And I'll say auditory cortex and hippocampus are very far apart in the brain. So this was really a way to study long-range coordination, or one example. So in this case, what Gideon did was to train animals to run back and forth for reward on this track. And 3/4 of the time, they got reward in an uncued way for running between these two arms. And then 1/4 of the time, a sound played. And then they got rewarded for approaching the sound, and they got a special reward there. And then later, Gideon was able to move the sound to the middle, and the animals did the same behavior. So we know they weren't just approaching the sound, they'd actually learned which way to go. Okay. Animals pick this up, so we can look at the inbound performance, which is from the outer arms to this home area, and that goes pretty well. And then after about 6 days, they figure out which way to go when the sound comes on versus not. Okay. So what Gideon did was to record this activity across many different sleep sessions and across different intervening run sessions. And I'm going to skip the sort of sound information. But the question he wanted to ask, which hadn't been addressed really at the time, was, is auditory cortex engaged in these memory processes? And if so, how does that work, right? And if we have our simple model of the hippocampus teaching things, then this is actually what the high filtered spiking or ripple activity looks like in the hippocampus. And the question was, does this propagate to auditory cortex? And very briefly, what we can do is look at all the neurons in hippocampus relative to these sharp wave ripple events. And then we can do the same thing for simultaneously recorded neurons in auditory cortex. And lo and behold, we do find that they respond, but moreover, we find that a number of them respond quite early, right? And so this is not consistent with the idea that the hippocampus is basically setting the stage necessarily. It sounds like auditory cortex might be talking to hippocampus, right? This is what it looks like on average. So this is the firing rate as a function of time. And you see the green in auditory cortex ramps up before the hippocampus. All right, and so we did a number of things in this study, but I just want to highlight one thing here, which is we wanted to ask the question, can we use auditory cortical activity before this sharp wave ripple replay event to predict what cells are going to be active? That is something like what memory is being replayed. And so we looked at activity in auditory cortex and asked how that predicted CA1 activity. And we found that even as we could go as far back as 400 to 200 to 400 milliseconds before the event. And lo and behold, what was happening in auditory cortex was predictive, better than chance, of what the hippocampus was about to do. Now, I'll just note these prediction gains are small, but this was a very old study with small numbers of neurons. So we think of these as kind of lower bounds. And then you see that during the event, this gets much more predictive. But critically, if we try and reverse the prediction, we get nothing. All right, so what does this mean? Well, it suggests that when these kind of memory processes are going on, maybe sensory cortical areas might be giving instructions on which important things need to be remembered. And then subsequently, the hippocampus might replay something which would broadly engage the rest of the brain. And lo and behold, then, if we ask after the sharp wave ripple event, we see that activity in the hippocampus here predicting auditory cortex later, that's a significant prediction. But auditory cortex during the event doesn't really predict hippocampus afterwards, right? So what we have then is this idea, we're going to come back to this updated model and then I'll finish up, that we have some sort of biasing activity that's flowing through this network in a way that we don't fully understand, but we have evidence that something is biasing stuff. And that might cause the hippocampus to replay a pattern, and that might then flow out to the rest of the brain. And so the idea is that maybe the cortical areas have traces of elements of experiences that are worth remembering. That biases what the hippocampus replays, and then that replay might engage the whole network and thereby bind different cortical areas together, bind the sounds to the sights and the smells or the task representations or everything else that makes up the full memory. All right. And so then again, in that replay, what we have is co-activation of these brain regions for that memory. Now, it actually gets a little more complicated than this. As it turns out, this whole circuit actually is also involved in motor learning. And what we see after this whole replay process has, and this is stuff I didn't show you, but we published a paper with my colleague Karanesh Ganguly earlier this year that looked at this. We actually see that maybe the prefrontal cortex starts taking over in the case, at least of motor learning. And after a lot of replay, we think that prefrontal and, for example, motor circuits come to be more connected and more coordinated during sleep. So with the idea that the hippocampus kind of hands off this memory process to the PFC with experience. Okay, and I just want to sort of emphasize here, oh my gosh, is this complicated? Oh my gosh, do we need to record from all of these areas in the context of these behaviors to understand how information flows? And we cannot do this in people. We cannot get the level of high resolution spiking activity that is actually underlying all these processes. So for me, at least, I think there's a real value in collecting and sharing these sort of large scale recording data sets that are now some things that we can start to record. Okay, so I'll stop there. Just want to thank the various people who contributed to this. And I'm happy to, in the last few minutes, take any questions if there are any.
I'll ask a quick question too. So how do you see this replay affected in sort of both ways of pathological direction, in the sense that if you have some model for rumination or PTSD, maybe is it that you're shooting that bias when it shouldn't be there, or if there are things, sleep disruption or things like that that are disrupting the replays, like what sort of the trajectory of that?
Absolutely. Yeah. So what we know is that, for example, in mouse models of Alzheimer's disease, the structure of these replay events is disrupted. So they don't have the same oscillatory structure. We don't think they engage the rest of the brain as well. We don't quite understand that yet, but that's the idea. And indeed, if you imagine a prioritization system that goes overboard, as in PTSD, we can tell a story about how those memories might come to be dominant, and those might dominate sensory processing. And there were some recent human imaging results, I believe, that suggested that during PTSD, the stimuli, the memories are processed as though they're actually happening at the time. So the idea would be that this is getting so ingrained in those networks that it becomes their dominant mode. But yeah, the hypothesis is that it's problems with these processes that indeed are leading to memory problems and therefore leading to the psychological challenges.
I see. We have one more question from Dr. Kilduff, actually. Is rat the preferred species for such studies, or can you take advantage of mouse Cree lines to identify specific cell types involved?
Yeah, great question. I'm a rat user because of the cognitive tasks that we use, and because it's very hard to get mice to do them and to carry all the hardware we'd like them to carry. But the majority of people in this field are using mice, and the technology for large-scale recording in mice has gotten much, much better with neuropixels and other things. So yes, there's a huge number of people doing sleep studies and other things like that in mice for exactly the reason you mentioned, is they can take advantage of all the cellular level specificity to try and understand the mechanistic components of this.